Hi guys, this is Kaylee. I tried to record this as a video where you could see me, but unfortunately it did not work. So I'm just going to present this slideshow just with audio, and I'm sorry it's not as exciting, but this way at least you should be able to see my pointer, which was not visible in the last one anyways. So I'm just going to present this talk kind of as a hybrid as I would to people in my department, but also explain a little bit more for you guys who might not be as familiar with biological concepts and graphs and whatnot. So the title of my talk is The Pilfering of Puffins, and I'm a master's student in the Cognitive and Behavioral Ecology program. So first off, I normally start with, can I go to the, there we go, okay, that was weird, sorry. It's a hard time switching slides. I normally start with acknowledgements, so I first off would like to acknowledge my supervisor Dave in my lab, and Story sits on my committee, research assistants who help me in the field, as well as Environment Climate Change Canada, which includes Greg Robertson, also sits on my committee, Dave Fifield, who provided a lot of help transport to my field site and also just help on the field site, and NSERC, who provided funding in my first year. So first off, what is kleptoparasitism? Well, it's the stealing of a procured food item from another individual, and this can be intraspecific, so between your own species, or interspecific between species. So in this picture here, you can see these two hyenas appear to be protecting this kill. Unfortunately, it was not their kill. They stole it from this African wild dog. That's a good example of kleptoparasitism between two different species of mammals. Now here's an example from the insect world where there's a fly here which is stealing resources from this praying mantis. It caught this paper wasp, and that's an example of kleptoparasitism as well. But probably the most common species and group that you see kleptoparasitism in is birds, specifically gulls and terns. I'm sure all of you guys have seen gulls fighting over French fries in a McDonald's parking lot. That is kleptoparasitism. They procured that food item. They didn't spend as many resources as they would if it was, say, a fish in a lake, but they did procure that food item. So that's what kleptoparasitism is in general. In my study system, my host is the Atlantic puffin, and they're a really common kleptoparasite host because they hold these large fish outside of their bill. They don't hold their food in their crop, which is a pouch in the esophagus that some birds hold. You'll see basically the birds that regurgitate food to their chicks hold it in their crop, but these puffins feed their chicks whole foods. You can see here in this video, this puffin has larval fish, which is smaller and less conspicuous, but it's still outside of their bill, feeding it to their chick in their burrow. Puffins are also really susceptible to kleptoparasitism because they come back to land to deliver their food. So they have to land on a steep marine grassy seaside slope and have to enter a burrow, which takes a little bit of coordination because the burrows are quite small, to be able to feed their chick. Now, unfortunately for the puffins, their kleptoparasite, the herring gull, is waiting. Herring gulls set up nests on top of the puffin slope. So you can see here, this is kind of what the slope looks like. There's different territories. This could be one gull pair, and that could be another, where they have nests, and they hold territories, and therefore they're there when the puffins are returning their fish. So what does this really look like? Well, it's a very physical interaction. The gulls sometimes make physical contact with the puffin and shake them up and grab their fish from them. And also sometimes the gulls just essentially chase or lunge at the puffins. But puffins do have a little bit of a defense. They have very strong bills and also really strong claws. And if you see here, this puffin is able to grab onto these, essentially the eye or basically the inner corner of the mouth of the gull, which is very sensitive, and bite down to essentially, if we anthropomorphize them a little, get back at the gull. But you can see these pictures where the puffin is quite a bit smaller than the herring gull, and therefore the herring gull is really dominating this interaction. So the herring gull has a risk of injury, but the puffin's risk of injury is much higher. The puffins also have that risk of losing their fish, which not only makes them spend more time and energy going back to the ocean to forage, but it delays the food delivery to the chick, which can be really important when chicks are younger because they don't have as many resources built up in their body yet. So my question really focuses on the behavior, specifically how do gulls choose which puffins to kleptoparasitize, and if puffins are sensitive to the victim choice of the gulls. So my study site was located in Gull Island, which is a part of the Willis Bay Ecological Reserve. It's a series of islands really close to St. John's. Gull Island is actually the largest colony of puffins in the Western Atlantic. And on this island, I set up little study sites across different portions of the island, which are indicated by these dots here. And in these study sites, I set up a grid of flags with known distances. So this is an example of flag as well as the one that's sitting beside me. Which I was able to use to calibrate a video for taking distances later. And so on this study site, I figured out where all the gulls like to hang out and spread non-toxic dye on their loafing or hanging out spots, which then they were able to passively transfer onto the gull, which the gull preened into itself and created individual markings. So this is what it would look like. I put dye a lot of the times on rocks because gulls like to hang out in a little bit higher of locations, but I also spread it on grass, and the gulls transferred it onto themselves. As you can see here, this gull has a nice big black spot under its chest. And in these two pictures, you can see different colors being used. This one, it has a blue spot on its upper chest, and this one has a nice pink line on its tail. And so with this dye, I was able to individually mark with unique patterns herring gulls across the island. It lasted for about a week or two, so I had to move across the island as the season progressed, so I didn't encounter gulls that I had previously marked that had been washed off. But I focal sampled about 73 focal gulls, which were 73 individual gulls on the island, which is quite good. My goal was 60. And so a focal sample consisted of me sitting in the bottom of this puffin slope in a blind with a camera. The camera was trained on these focal gulls, so ones that were dyed, and the flags that I used provided a little bit of a circle with my focal area, which was about a 4-meter radius around each focal gull. And I recorded them for 30 minutes and told the camera, dictated anything that happened in that area, how many puffins were bringing back fish, what happened to the puffins, if the gulls attacked any puffins, the outcome, basically any variable. To be able to use. And this is the framework that I use, where there's two separate sections. There's one where it's completed landing and two where there's an aborted landing. And the puffins usually, basically all my variables are binary. So there's a yes or a no. So were they attacked or not? Did they lose their prey or not? And they all end up essentially returning to the ocean to forage after either losing their prey or being able to enter their burrow and feed their fish. Everything gets reset when they approach the island again, and the entire process starts over. They're able to choose a different strategy than they chose last time. Using this framework, I have a few video examples to help you guys visualize this a little bit better. So in this video, you can see a puffin flying in with large fish. It lands in the circle with these two very large fish on the slope with a herring gull right here and also right there, and has to run to its burrow. This case, he successfully makes successfully makes it in and is able to feed its chick. Will have to return to the ocean to forage because it successfully fed its chick. But in this case, it was a win for the adult puffin and the chick. In this case, the gull won. So the puffin, again, completes its landing, so that part's the same. But you'll see here the gull lunges out and makes physical contact now with the puffin's head and kind of shakes the puffin. You see the puffin fly off now with no fish, and the gull will reach down and pick up the puffin's fish and consume it. Now, gulls are crop feeders, so this gull could have used this to provision its chicks later, but obviously I'm not sure. That puffin then had to return to the ocean to forage. Again, it will ultimately reset at the next approach. So for a second example, here's an aborted landing. So you see this puffin flying in here. Aborted landings happen when a puffin approaches the slope but ends up slowing down and turning away distinctly. In this case, this puffin was not attacked by this herring gull here. And so the puffin is simply able to approach the slope again, and it's much quicker than having to complete the landing, either losing their fish or having to take off and approach again. So here's another example, but this aborted landing ends in attack. So this puffin flies in here, this gull jumps out, tries to make contact, doesn't end up making contact. The puffin keeps flying and will be able to approach the island again with its prey still in its bill. So with that in mind, those are my variables. And what I did in the lab was I uploaded all my videos, which is this right here, into a behavioral analysis program called Boris. And I used the markings that I had on the slope. So right here, there's one flag, and I also used natural markings like this tree here. and calibrated these little circles that I have here. They're a little harder to see, but these right here, calibrated to both the horizontal measurement, in this case it was 3.3 meters, and also the vertical measurement, which is between these two flags. Because I was recording a slope, it was not a straight wall, and so there was perspective issues, so I needed to account for both horizontal and vertical measurements. So this is why, because this large circle, they're both calibrated to 8 meters, but this large circle is my horizontal measurement, and the smaller circle is my vertical measurement. So this 8 meters is the same as this 8 meters. And so my focal area, as you can imagine, was not a circle, it was an oval. And so in this area, I listened to my dictations and transcribed anything that I said happened, but I also was able to transcribe any interactions that I missed in the field, and which is one of the big benefits of recording videos, not having to rely on your reaction time in the field, because there were definitely quite a few puffins that I missed coming in, and my video were of sufficient quality that I was able to see fish in their bills as they were coming in. So with that in mind, I'm going to get into my results a little bit now. I'm going to give you a really quick crash course in my Mosaic plots, which is how I present my results. They are a visual representation of a contingency table, which is a table where there's two variables and each of the options are yes or no. So in this case, we have survival and treatment as just a mock example. And we can envision this square as the total sample size, so the total number of individuals that you observed. And we can first separate it by one variable. So we'll separate it here by survival into alive individuals and dead individuals. As you can see, the size of the box indicates how many individuals essentially were part of that category. So many more individuals died than survived during this experiment, this mock experiment here. And now if we want to see how treatment relates to survival, we again separate it by treatment 1 and treatment 2. And you can see the difference there. This box is a lot larger than this box, which indicates that many more individuals stayed alive in treatment 2 as compared to treatment 1. And obviously the inverse is true for the alive and dead. So this is how I present my results, and it's pretty intuitive, but it just takes a little bit of an orientation. So, first off, what puts a puffin at risk? Are puffins flying in with no obvious fish at risk, or are these puffins with very large obvious fish at risk? Gulls should not attack puffins that don't have an obvious fish. So I related gull victim choice to the bill load of the puffin, and you can see here that 46 out of 46 attacks happened when a puffin was carrying a large conspicuous fish. It could also be larval fish. In their bill as compared to this one. So essentially the profile of the puffin showed that there was a fish in their bill. Now I was interested if puffins were sensitive, so I related puffin landing choice, so aborted or completed landings, to this bill load. And you can see here that many more puffins aborted their landing when they held conspicuous fish as compared to inconspicuous fish, so they were sensitive to the presence or absence of fish in their bills. Next, I was interested in looking into focal gull direction. It's a little bit of a weird variable, but basically if the focal gull was facing in the direction where the puffin either landed or aborted landing, basically if the gull observed the puffin's landing or aborted landing, was it more likely to attack that puffin? And in the field, I thought yes, and so I wanted to test that just to double check my assumptions, and I was right. 45 out of the 46 attacks that I looked at happened when the focal gull was facing the puffin at the time of puffin approach. It was very significant, 0.0005. And then again, were the puffins sensitive? Did they change their landing choice in accordance with focal gull direction? And no, they're not. If you know anything about stats, 0.9 is very, very, very insignificant, almost as insignificant as you can get. There was no difference. Puffins did not abort their landings or complete their landings any differently in accordance to the focal gull direction. So that was interesting. Next, I wanted to know if gulls were sensitive to puffin landing choice. So, as you can see here, with my cursor, there we go, 29 out of the 46, I don't know. Anyways, 29 focal gulls, 29 attacks happened when the puffin completed their landing as compared to aborting their landing. And as you can see, the sample size between both are essentially even, so it wasn't an issue of different samples. So yes, gulls are sensitive to puffin landing choice. Then I was interested in if prey size had any impact. It's expected that gulls will like to attack puffins with larger, more conspicuous fish more often because they have a higher calorie content. But it's also possible that they're just more conspicuous and easier for the gull to spot. However, birds have really good visual acuity, and so it's expected that the gulls would definitely be able to pick out this larval fish, especially if I could pick out the larval fish. But larval fish essentially don't have any fat content, and they're not great food. Will keep the chicks alive, but it doesn't let them put on very much weight. So again, is the victim choice dependent on puffin prey size? As you can see here, 34 of the attacks happened when the puffin was carrying adult fish, as compared to 6 with larval fish. So the gulls were preferentially targeting puffins that held large fish as compared to larval fish. So again, do puffins exhibit a sensitivity to this? Do puffins change their landing choice in accordance to the prey size they are holding in their bill? And the answer was no. It's a little bit less clear here in this graph, p-value of 0.08. My alpha level is 0.05, and so it's greater than that, and therefore it's insignificant. This graph is a little bit misleading because it's essentially not fully representing my models, because my models also take into account individual gull ID. So it would look a little bit different, but mosaic plots are simple and hard to incorporate generalized linear mixed models into. So basically, based on this, puffins do not abort their landings any more often than expected when they held adult fish as compared to larval fish. And lastly, I was looking into aspects of puffins' landing that impacted their essentially risk of being kleptoparasitized. Now, I'm only presenting this one variable, but I do have other ones. They just were really not significant, so not interesting. So is there a difference between the distance between the landed puffin and the herring gull, and is that related to the herring gull's victim choice? As you can see here, yes, the puffins that got attacked were landed much closer to the herring gull as compared to ones that were not attacked, and it's capped at 4 meters because my focal area was 4 meters. Obviously, there's no upper limit to the no attack because a puffin could land across the world in Europe and not be attacked by my focal gull, so it's a little bit kind of weird in this category, but it is very distinct, it is very significant. They attack puffins that land closer to them. With that, I'm just going to conclude by reiterating that gull victim choice is based on both prey size and presence, the observation of a puffin's approach, the puffin's landing choice, and the distance to a landing puffin, where puffins are really only sensitive to the presence or absence of prey in their bill. Now, this can be due to different interests between the puffins and different strategies. Puffins might be more willing to land when they have a large fish, even though they're at higher risk, because that large fish has more benefit to their chick, and puffins are only attacked not every puffin that lands is attacked. The attack rate is possibly low enough that some puffins are willing to risk this, and the difference between individuals might be swamping the difference in the strategies. It just basically makes it harder to tell the difference, and there's no way to account for individual puffins because there's hundreds of thousands of puffins on the island, and I wasn't able to individually mark them. So unfortunately, I'm not really able to answer individualized questions for the puffins. But regardless, the gulls seem to really be honing in on a lot more factors than the puffins. And finally, I'm going to try to work towards understanding the full story, which includes pulling into the story information about gull success and puffin retention, and if there were any strategies that changed either of these. Spoiler alert, there are none, because I have to take into account individual ID for my gulls, which makes my sample size a lot smaller, and can't use the normal stats, and it's a long story, but essentially there's really nothing that impacts the gull success and puffin retention. I just didn't want to get into that for this talk, for my public talk for the conference talk that I gave yesterday. I also only had 12 minutes, so as you can see, I'm probably over. But they didn't change their success or retention didn't change really depending on the types of strategies the Puffins or Gull used, which is a little bit disappointing, but it's interesting nonetheless. So at this point in my talk, I normally ask if anyone has any questions and thank everyone for listening. But if you guys have any questions, obviously you can't ask me right now because you're just listening to a recording. So you can either email me or maybe we can do a video chat. And if you have any questions, I can try to answer them for you. Thank you.